Next, we have Cadex Genomics. Cadex Genomics helps molecular diagnostic tests to guide cancer treatments using liquid-based technology to provide personalized insight into the therapy response. By combining proven approaches to measuring DNA with cutting-edge technology to improve accuracy and sensitivity, Cadex Genomics provides unique insight into therapy response using simple blood draws. Alibrex is a blood-based assay developed to help physicians identify patients who are not responding to drug therapy. The test is optimized to maximize NPV, that is, minimize the number of false non-responders. Dr. Sinha, Sudhir Sinha, the CEO, holds the position of adjunct professor of biochemistry and molecular biology at Tulane University School of Medicine in New Orleans. Dr. Sinha has over 36 years of experience in biochemical research, cancer biomarkers research, and human DNA genotyping and assay commercialization. Previously, Dr. Sinha founded Genomics and was also the founder, president, and lab director of Relia Gene Technologies. As president and chief scientist of Relia Gene, one of Dr. Sinha's major achievements was designing and producing the first commercial YSTR DNA testing kit. Dr. Sinha also served as the lab director of Relia Gene's CAP-certified CLIA laboratory. Dr. Sinha was awarded an export promotion award by the U.S. Department of Commerce for the worldwide export of this novel DNA test kit. Dr. Sinha built Relia Gene from a 2-employee company into a 90-employee company and successfully sold Relia Gene in 2007 to Orchid Cellmark, which is now part of Laboratory Corporation of America. Dr. Sinha earned his Ph.D. in chemistry from the Indian Institute of Technology, Kanpur, and completed his postdoctoral fellowship in biochemistry and molecular biology from the University of Miami, Florida. So we're very happy to have you. Thank you.
Okay. So thanks everybody. Thanks very much, Chad and organizers to give me this opportunity to present. And as you heard, I'm in New Orleans. This company is born and bred in New Orleans, where we are. Of course, we have partners to raise funds in California, and some of my partners, and I'll talk about it later. So the basic things about this is very simple assay. It's a very simple assay. It's a qPCR assay to look at the cell-free DNA in the plasma. So, and I'll go into difference. What is the normal other ctDNA assay and what is our cfDNA assay. This is a cell-free DNA assay. So the next slide. Sorry. Okay, disclaimer. Okay, the current status of test is what is the unmet medical need. Now, unmet medical need to evaluate whether a treatment is working or not working is to look at the CT scan after several rounds of chemo after 3 months. Some places in more than or some places less than, but average on 3 months. So for 3 months, the patients don't have any idea all these drug which he is given to, or including immunotherapy, whether it's working or not working. We have tried to find a solution. So this, I put it in break in several parts. So this is the way the CT scan is done, the first treatment, and after that the second round, third round, and in the meantime the patient's cancer is growing if it is progressive. The cancer antigen tests are available, but they are not actionable because CEA, PSA, those tests everybody knows, is give you an idea. If changes rapidly, then oncologists but cannot take any action based on that because they are not age specific. So we have developed an assay, a test, which we call Librex. And this assay is based on, as I said, cell-free DNA. God, this screen is, I need to increase the size of the screen. No, it won't do it. I don't know. It's so small here. So, okay, yeah, that's much better. Thank you. Okay. So the assay basically, let me say about the design of the assay, is basically we take 2 blood samples, first before the treatment starts and after the treatment start, after about 15 to 21, 30 days. So it coincides with the cycle of treatment, like the patient comes in, they get the first dose of chemo or other medicine, and then they go back and come back again, and before they get the second dose of treatment, we can have a blood draw, and that blood draw, the 2 blood draws is the changes in the cfDNA which we are looking at. So it's a very simple assay in that sense. See the first trial we did, I have to come this way. Sorry about all this. Because the first trial we did was, and I'll talk about it, but in this case, and the results I'm going to present here, is the blood was shipped from 15 different centers to New Orleans. So it's more like a real life assay if we want to do as an LDT. So there is a cancer therapy monitoring the market potential here, and this is an estimate by our team, marketing team, that there is a big need, because right now the patients undergo, and this is very critical, and if anybody have seen, one of the founder of this company was himself and was an oncologist, Gary, and Gary himself was a cancer patient, and he saw me present this work in an ASCO meeting, and he came to me and he said, this is very useful, because I went through so many unnecessary treatment, and those treatment made more harm than good to me. And he was instrumental to put me, he said, you need investment, you should form a company, and he was instrumental putting in touch with other partners. I'm a chief scientific officer of the company. CEO is Bill Hack, who was with a Genomic Health before. So he was one of the marketing director for the Genomic Health before. So he knew, and we put together the team. So the treatment, unnecessary treatment affecting the patient, that was his biggest concern. He was very good. Unfortunately, last year he died of the stage 4 cancer before I could bring it this assay. So there are other ctDNA, cell-free DNA assays by different lab. Natera is one of the leading one, but they are all looking at the mutation. So one of the assay I just point out is take the solid tumor, do the sequencing, find the targets. Of course, that is very useful because then you can find the targeted treatment. But for the monitoring, then you look at the mutation and then you follow the mutation. The problem is many times there is a clonal change in the cancer, and it's not the same mutation. There is new mutation rises out of there, and that makes the monitoring difficult. Another point, and I'll bring it later, that the assay sensitivity, analytical sensitivity of the assay. So what we have here, that in cell-free DNA, and it's all well known, that the cell-free DNA sheds small fragments of DNA in the plasma. When you look at the plasma, initially when I was trying to see this, I saw a NIH, NCI solicitation. It was a contract solicitation looking for cell-free DNA kind of assay. My field, as you heard, I formed a company called Reliagene here many years ago, and I was doing diagnostic as well as forensic DNA testing. I was fortunate to be associated with Tulane. I came in Tulane as a professor in the first place. And one of my colleagues, Prescott Denninger, has done a lot of work in the retrotransposon area. And I met with him many years ago, almost 23 years ago. He put me in the work with retrotransposon. And then another colleague, Mark Batzer, who is in LSU Baton Rouge, he is also working on ELU and retrotransposons. So I collaborated with both of them. I have published a lot of papers together, and a lot of patents came out of that work. I got my first grant from National Institute of Health on retrotransposon in 2000 to try to look at these small fragments of DNA and identify the either human identity or quantitate the DNA, those kind of applications. So I have many patents, about 13 patents awarded in that field. And I saw this solicitation. One of my colleagues, Jonathan, is sitting here, and he said, why don't we apply for a grant? And I said, I haven't got grant in cancer field for last 20 years, 30 years. Now just apply a grant. A lot of people has put so much work into all the time. I knew how much other colleagues in Tulane work in this area. There is no chance. But I still said, look, we have a technology. We already have a technology where we can quantitate and identify small fragments of DNA. And I developed the technology for the forensic application. But why don't you apply for that? So we applied for a SBI grant. And I was so fortunate. First time applied, we got phase 1, phase 2, and got over $1 million money. And that's how I started this company and started this work. So this was small fragments of DNA in the blood. And as I told you, the technology is you take the 2 blood samples after 21 days apart. You see that initial treatment happens. There is a lot of cell-free DNA because of cell death of the treatment. So you have to give at least 4 or 5 days time frame before the blood's level of cfDNA comes back to normal state. So this is the retrotransposon, ELU retrotransposon. I told you this story. The beautiful thing about this particular marker is, and by the way, ELU name was given by Prescott. He gave the ELU name for this because he used ELU1. I asked him, he said, ELU1 enzyme to cut the DNA to look at the fragments. So this particular retrotransposon is present in every chromosome, all the 23 chromosomes, and in many different locations on the chromosome. So it is a whole, if you look at that particular sequence, DNA sequence, it is present in the whole genome, and it gives you the whole snapshot of the genome fragmentation. That is the beauty of particular this target, which is the patented target by us. So this one, at this particular slide is Mark Batzer's publication slide. And then there is many people have seen nowadays, not only cfDNA, because ctDNA, I'm separating those two, cell-free DNA and cell-free DNA, where you are looking at the tumor mutations, ctDNA. ctDNA assays are there, many, because it's much specific. You are looking at a KRAS mutation or others. But that, as all the patients don't have that particular mutations. That can happen difference. So you cannot follow every patient. In cfDNA, you can follow every patient. But the biggest criticism I faced initially when I started it, and of course I overcome in phase 2, first round, we got that. When I applied that, we didn't overcome that in the phase 2, was that how do you separate normal cell death cfDNA from the cancer cfDNA? So cancer cells are producing cell-free DNA, normal cell deaths are producing cell-free DNA. How do you separate that? This was the biggest criticism. And then I later found out with many publications, this is one of the publication data, is not my data, where they look at the pancreatic cancer and they sequence the cancer DNA, cfDNA, and they found the mutation containing cfDNA is mostly smaller fragment than the cfDNA coming out from the non-cancer cells. Like a wild type, they looked at the same place, the wild type sequence. So wild type sequences were higher fragment than the cell-free DNA coming out from the cancer cells. So that is the fragmentomics we are talking about. Analysis of fragment analysis of looking at the cell-free DNA and making it cancer specific. So cell-free DNA, which is total all the small fragments of DNA, now you are looking at smaller than 100 base pair fragment of cancer cells, 105 actually in my case. Then if you look at less than 105 base pair fragments, then you make it more specific to the cancer cells. Now you filter out the effects of the white blood cell death, which happens in many cases during transportation, even in the extractives. So that's the technology. It's a simple qPCR technology and using this target. So the first study, and I was fortunate here, got introduced to MD Anderson, Dr. Scott Kopech. He's doing in CRC, colorectal cancer. Scott, when we talked to him, we went to Houston and talked to him and showed him my assay result. I got some cancer patient samples from Jewish General Hospital in Montreal, and that showed that the cancer patients' cfDNA, small fragments were higher, normal patients were very low. Those data I had, I presented to MD Anderson, and Scott said, okay, I'll believe you if I send you some blood samples, 32 or few blood samples. You don't know anything about the patient. You do your assay, send me the result, and then let me look at it. So he sent me 32 patient samples. Out of that, we did the cfDNA assay, 80 base pair at that time, single 80 base pair fragment quantification. And the quantification we do in qPCR with a standard curve. The standard curve is calibrated, DNA is calibrated against the NIST DNA. So NIST DNA, what they said, DNA quantifications, National Institute of Standards, that's how we quantitate relative quantification with the NIST DNA. Send him the result, and this was the result. The 100% specificity in predicting if the cancer were progressive. What I said, the cell-free DNA small fragments levels were increasing. They were all progressive cancer, 3 months, 6 months, followed by the CT scan, actually RECIST measurement. So it went 100% specificity and about 60-65%, because there were some patients who initially the cell-free DNA level went back down and then immediately went back up. And those patients, if you look at, we are looking at a snapshot of the initial 21 days, those patients were showed. So, but the patients whose cell-free DNA small fragment increase in the first 21 days, they continue to grow. The cells continue to grow. So this was the presentation I did in there in ASCO meeting. Actually, MD Scott Kopech's groups did in ASCO meeting. And then we have the Cadex Genomics. We raised some funds, first round with friends and family, actually mostly oncologists, who funded it, where we did the trials. And we have 130 patients, all stage 4 patients, but they are in 3 different cases. You see lung, 50% of the lung cancer. They were undergoing all type of treatment, not one treatment. So it's treatment agnostic, mutation agnostic. So they're going all type of treatment, small cell lung 8, breast cancer 21, and colorectal cancer 51. This is the total patient population we got from different places. Blood shipped to New Orleans, and I analyzed those results. And this, after the analysis of the result, I told you less than 105 base pair fragment, and is a change in the concentration between the 2 time points. That's what is the parameters. So we gave it one of our partner. He's a professor at George Washington University in St. Louis. He's a biostatistician. He looked at the data and he analyzed the data during, actually, this was very impressive when he told me that Bill Shannon, he took bootstrap and he explained me the bootstrap resampling was to show what happens instead of 130 patients, you do at different populations and different range and different area. Will this model, and created a model to measure the cutoff, if this model will hold. And with all the, it still was the positive predictive value was 99.3%. Okay, that's what we are focusing on the identifying the progressor. So not focusing on the sensitivity, like whatever going down, focusing on the identifying the positive predictive value. Because if oncologists say to take the treatment, off treatment that patient, we should be absolutely sure the treatment is not working. So is not looking at the average. We put the cutoff very high. So that's what, and this is the AUC curve. But you see there, that side, the scatter plot there, all the positive progressive samples are there in the top. Then there is few in the middle. Actually 60-65% we could put the cutoff, but here put 90% there to cut off to make sure we have a good result which can be relied on. So this is where we are. This result is pretty exciting. We just got this result and analyzed it, and we are now raising some fund to go further. So the therapy monitoring is the main application, but we can go to non-adjuvant setting also. That's expand it. It can be used that monitoring the untreated stage 4 patient disease progression and also surveillance. That's the idea in the future. But right now we are looking at stage 4 cancer patients because we can look at the endpoint quickly. We can go much faster to check the application. So the goal is that right where we are, right now we got our formula, we got the initial clinical result, but we need to validate, as you heard talk before me, to do another 140 patient blindly, apply the formula there, and see that we can predict all of them correctly, that they are progressive, and then go for a Medicare. One of my team member went and talked to MolDX, contractor for Medicare, and they were very interested. They said, this is very good. This is payer-focused test because you are not giving unnecessary treatment. So this one is a patient and payer-focused test. So you don't give unnecessary treatment. So they are very interested that yes, this will be. There is actually already Natera has got already a LCD issued for ctDNA, and this can come under that category. So that's what we have that we are looking at. So that's the main the CV clinical validation, and then we can have the LDT test, set up a CLIA lab and set up. So that's what we are. We are talking to several people. There is a partner, a strategic partner, ready to sign up so that they already have a CLIA lab which we can join hands with and then do this second page of study. This is our team. Bill Hack, as you say, he was in Genomic Health. He is the CEO of the company. He is the one, I credit him to raising funds so far. About $3.5 million so far we have raised. And that's what this study and clinical trial we did. And then he's our chief, after Gary's death, Kevin has joined as the chief medical officer. He's in San Francisco. He's a practicing oncologist. He believes in treatment holidays, and he believes in there is too much treatment going on. We should control that. Unnecessary treatment should be reduced. And then Patrick was one of the founder of Genentech. He's a board member. He's also an initial investor, and a founder of this company. And Bill Shannon is the biostatistician who did all the biostatistical analysis. He has his own company called BioRankings after he retired from there, and he does all this kind of diagnostic and clinical assay biostatistics. So he understands the field very well. The unique differentiation we have that it is a patented technology, IP is there. We have PCT outside the country also, many places. In fact, we have applied with too many places. I might have to cut some down. It's too expensive. And so that's where we are in there. And this is our contact information. Thank you so much. If there is any question. The key here is really very simple assay, very quick turnaround time. 2 to 3 hours total assay time. We need a very small amount of blood, total 1 ml of blood. We can do duplicate assay. Each assay I use only half ml of blood. With Scott, that's why Scott was able to spare that. He said, oh, you need only half ml of blood. I can send you half ml of blood. That was left over from his other studies. So we can do that. That's what we are talking to somebody else or to another strategic partner who also are in the liquid biopsy stage and said we have a lot of blood sample of the other study. We can probably get that and do the clinical validation without doing prospective collection, but we don't know yet. We are just in talks. So very small amount of sample, and the main reason for that is very high copy number of the ELU retrotransposons. 1800 copies per cell. So our analytical sensitivity is less than 1/10 of a cell DNA. Yeah.
You said it was treatment agnostic?
Yes. Oh, no. No. We did do some, I didn't present it yet, we do some patient where there is chances like, radiation, cell death, or some other like myocardial infarction. Between we looked at that. So there were 3-4 indications this assay will not be valid.
As regards prostate cancer, is hormonal therapy, is it associated?
No, prostate we didn't do, but immunotherapy on lung cancer is a lot of patients. And immunotherapy is even more interesting. Lung cancer is even more interesting because immunotherapy initially in CT scan, there is a pseudoprogression. The cells go around the tumor and attaches to themselves. And when you do the CT scan, it looks like the tumor grew, where the tumor may be dying. So it's a false result on CT scan. And we looked at it, and the CFTNA levels went up. There were some CT result which shows increase, but our assay did not show the increase, and they were not the increase. So that's okay. Thanks. I want to give credit to Jonathan, who is here. He is the one who pushed me to write the grant application and put the grant application together, really. Otherwise, I was not thinking to apply in cancer because I was not in that field for last 20 years. I was one of the one initially in the field, and as you heard, I isolated GM-CSF for the first time, and actually Amgen gave me a contract to supply purified GM-CSF, and I did, which is now a drug, the Nepogen. So I was one of the first purified, but we didn't patent that in those days.